The Hong Kong Polytechnic University, PolyU, leverages its world-class academic and research excellence to develop innovative solutions for a more sustainable tomorrow. In July 2022, the PolyU Academy for Interdisciplinary Research PAIR was established as the university's central research platform, dedicated to providing world-leading interdisciplinary solutions to address significant societal challenges. PAIR is composed of multiple constituent research institutes and research centers. Each research unit focuses on a particular research area and is joined by researchers from various PolyU faculties, schools, and departments. PAIR research areas include advanced technologies and manufacturing, good health and well-being, and smart and sustainable cities. PAIR researchers develop new materials, systems, technologies, and techniques that support industrial modernization, upgrading, and transformation, look into the factors and mechanisms influencing human health and wellness, and develop evidence-based solutions, and generate smart technology-based solutions for making urban operations and services more efficient, connected, and eco-friendly. We particularly value impactful collaborative activities in research and scholarship. PAIR researchers pursue research collaboration at different levels. In addition to interdisciplinary collaborations within and between research units, PAIR and its units establish partnerships with industries, governmental bodies, universities, and other external stakeholders. With internal and external collaborations, we have successfully translated PAIR-led innovations into commercializable solutions and practical applications, and established joint laboratories and research entities with external organizations, driving scientific breakthroughs, innovation and technology development, and socioeconomic prosperity. PAIR's research excellence and research impact have been widely recognized at local, regional, and international levels. Our research outcomes have been published in leading journals, including Nature and Science. Innovations led by PAIR researchers have won prizes at various competitions and events, including the International Exhibition of Inventions Geneva. Many PAIR researchers have been named among the world's top 2% most cited scientists in their own areas of specialty. Individual researchers have been selected to the National Academies of Science or Engineering and as fellows of professional associations, and have been bestowed with local, national, and international awards and honors. Harnessing the strengths of PolyU's research and world-class scholars, PAIR will continue our unwavering efforts in research and scholarship, knowledge transfer, and collaboration to develop impactful solutions that benefit Hong Kong, the nation, and the world.
Okay, so good afternoon, and Professor Boehm, and ladies and gentlemen, welcome to the PAIR Distinguished Lecture. So I'm Xiangdong Li, Director of Research Institute for Sustainable Urban Development, ISUD, under our PAIR, PolyU Academy of Interdisciplinary Research. Okay, it's my great pleasure to introduce today's distinguished speaker, Professor Alexandria Boehm from Stanford University in the United States. And Professor Boehm and holding multiple appointments at Stanford University, serving the Richard and Rhoda Goldman Professor of Environmental Study and the Professor of the Ocean and a Senior Fellow of the Woods Institute for the Environment. And Professor Boehm's research has a major implication for both people and planetary health. And the work examine and how pathogens move through the environment and also reach people in both developer and developing countries. Affecting our advance our understanding of how infection spread through water, sediment system, and contaminated surfaces. So she also worked extensively on like coastal water quality, marine ecosystem problems, pollutions, studies, passaging, facial like nitrogen dynamics, and the use of environmental DNA to monitor the marine ecosystems. So one of Professor Boehm's most influential initiative is a wastewater scan. So that's a national program directed the use of wastewater surveillance in direct and tracking the spread of the infected disease, including the COVID-19 and other influenza and other disease. So this work have demonstrated this kind of importance of the wastewater monitoring for public health, making about particular for this served community, both in the US and other places of the world. So with the true distinguished, Professor Boehm is very much on the multidisciplinary research of her research, her study, which bridging the environmental science engineering with urban wastewater system, microbiology, public health, policy, and climate science. So she also served extensively on scientific and policy advice bodies related to coastal water quality, ocean protection, and environmental health. Professor Boehm's contribution to promoting public health from environmental transmission, infectious disease have earning widespread recognition, including election to the U.S. National Academy of Engineering, among many other science engineering awards. She's also a senior fellow of our PolyU pair. So without further ado, I would like to welcome Professor Alexandria Boehm to give us a distinguished lecture now.
Thank you, Professor Lee. Hi, everybody. I'm happy to be here today. And I've been in Hong Kong less than 48 hours, but it's been a lot of fun so far. So today I'm going to talk to you about influenza in wastewater and how we can use it to infer information about human and animal health. So a brief introduction to influenza. It is a leading cause of mortality and morbidity in the world. Annually, there are 650,000 deaths and 1 billion cases. In fact, many of you in this room probably had the flu this past winter. Unlike most human viruses, it has really broad host tropism, meaning that it can infect a really wide range of animals, including humans. And it's designated by different subtypes like H, X, and Y. You might have heard of H1N1, H5N1. It is an enveloped, segmented, single-stranded RNA virus. So its genome has multiple little pieces in it. And it has, because it's segmented, it can have a lot of reassortment events and mutation events. And so it is considered to be a virus of high pandemic potential because it can acquire new instructions in its genome and become more virulent and also be passed among different animals. Transmission modes vary by host and subtype. It can be transmitted by air, by water, and by fomites. And this little map here shows some transmission pathways that involve birds, but all these different animal silhouettes that you can see here can be infected by influenza A. So today I'm going to talk about wastewater monitoring for infectious disease surveillance generally, and then talk about where we find viruses in wastewater. And then I will talk about the evidence that we've collected about wastewater monitoring informing the spread of influenza A. And I'll talk about our experience. And finally, a little bit about metagenomic informed influenza monitoring in wastewater. So hopefully I can get through this in 45 minutes. Okay, so first, just a brief introduction to the topic of wastewater disease surveillance. Many of you may be familiar with this. The idea is that influenza, in particular disease surveillance, is really impacted negatively by the severe delay in getting test results and the fact that most people don't get tested when they get the flu. So only the most severely ill people go to the doctor. So this sort of surveillance misses most influenza infections. And it also misses emergence of new subtypes. So because people aren't tested, we don't know which subtypes are circulating necessarily in the population, or if our vaccines will protect against the circulating subtypes each season. So wastewater represents a biological composite sample of the entire community. Everything that goes down our drains ends up in wastewater. It might be sputum. My marker was going all over the place. So when we brush our teeth, what goes down the drain, what comes off our skin and off our body in the shower, and what goes down the toilet all ends up in the wastewater treatment plant. And we know from the very limited work on influenza shedding and human excretions that its nucleic acids can be detected in both feces and sputum. So this is some data from a study. One of my students did a systematic review and meta-analysis of the concentrations of influenza A in human excretions. There's surprisingly little data on it because clinicians don't need that information to diagnose the flu. They just need presence or absence in a swab, for example. But these studies show that we might have 10 to the 5th or 10 to the 4th copies of influenza RNA per gram of stool. And for sputum, it could be higher, 10 to the 6 copies of influenza RNA per mil of sputum. So we have reason to believe that this might end up in wastewater. We might be able to detect it in wastewater based on this kind of data. And we can detect, we can use wastewater from a bunch of different places to do disease surveillance, including a wastewater treatment plant. This is a map from the Bay Area showing different plants we were working at. That's me at a wastewater treatment plant. I love to visit them. Sub-sewer sheds, portions of a sewer shed if we sample from a manhole, and then actual school or buildings. We can go in and sample the pipes right there if we want. Okay, so the next question is, where are the viruses in wastewater? This is a picture of a wastewater sample. It is a complex mixture. It contains liquids and solids. The solids can consist of particles greater than 0.2 microns in diameter. They may contain bacterial cells, fecal matter, mammalian cells, food waste, etc. Very complicated matrix. Some of our earlier work with actual wastewater samples suggested that there were higher concentrations of viral nucleic acids in the solids compared to the liquids. So these data here are of Mpox DNA. So this is a virus that causes monkeypox. These are actual wastewater samples from San Francisco. This is from a plant called Oceanside. This one is from a plant called Southeast. And the units here are copies per gram of Mpox DNA or mil. So a mil weighs about a gram. And the triangles are the concentrations in the solid fraction of wastewater, and the circles are in the liquid fraction. And you'll notice that the liquid fraction has about 1,000-fold lower concentration than the solids. So we were observing this in actual wastewater samples. So one of my students did some viral adsorption experiment. She took a bunch of wastewater samples from wastewater treatment plants, spiked them with a whole bunch of different viruses, mixed them till the solution came to equilibrium, separated the liquid from the solid phase, measured concentrations of the nucleic acids of all the viruses using droplet digital PCR, and then did different adsorption models to figure out what is the absorption coefficient for these different viral nucleic acids. And what she found is that for all these different viruses listed over here, so dengue, hepatitis A, influenza, SARS-CoV-2, West Nile virus, Zika, that the concentration on the solids, which are on the y-axis, were always much higher than the liquids on a per mass basis. So this is the concentration in the liquids in copies per mil, concentrations in the solids in copies per gram. So if you draw a line here, so it's 10 to the 6, a million copies per gram, and then it's like 10 to the 2 copies per mil in the liquid. So for all these different viruses, the adsorption coefficient was over 1,000 mils per gram, suggesting that all these different viruses have, the tendency to sorb to the solid phase of wastewater. And here's some actual influenza data from a real wastewater treatment plant. The influent, or the liquid here, is all non-detect basically. And the solids are like 10 to the 4th copies per gram. And the liquids, again, are non-detect. So actual wastewater also showed us that we had very high concentrations in the solids. So with this information in hand, we have done a lot of research on using the solid phase of wastewater to do wastewater surveillance. Here are some of the papers that we've published, including one in Mpox, on all these different respiratory viruses, arboviruses, dengue, HIV, and also different enteric viruses, and also SARS-CoV-2. So all this work has been done with the solid phase of wastewater. And I'm going to talk to you more specifically today about influenza A. But first, I'm going to talk to you about our project WastewaterSCAN, which is where these data come from. So during the pandemic, we scaled wastewater surveillance across a bunch of areas in San Francisco. And then a very generous donor wanted us to scale the work across the United States. So we then scaled this to the entire country, and we work with 150 sites across 40 states. And these purple dots show all the different locations where we work. So all these wastewater treatment plants send us 3 samples per week, and we process the samples within 48 hours, and we post them to a public dashboard. So our public dashboard is at data.wastewaterscan.org. And these are all the targets that we monitor for. Results are within 24 hours. There's a lot here, and I don't think I'll read them all. But I will say that I could have created a talk today based on any one of these viruses or fungi, but I am focusing on influenza A for this talk, which is in red up here. And then I'll mention briefly influenza B, RSV, and human metapneumovirus. But we have papers on all of these different targets, and we continue to monitor them now on our dashboard. So the measurement techniques we use are, we focus on the solids in wastewater. So we get our samples, we dewater the samples and resuspend the solids in a buffer. We do nucleic acid extraction and inhibitor removal and droplet digital PCR, and we do lots of multiplexing with the PCR assays. Okay, so when we started this work on influenza, there was maybe 1 or 2 papers that had been published where they had reported that they had detected the presence of influenza A in wastewater, but they were sort of like one-off studies. And I remember a colleague submitted a paper on this topic, and actually the reviewers didn't believe that it was actually influenza. They thought it was contamination. Nobody believed it. So there wasn't a lot of evidence that we could do this kind of work with influenza. But Krista Wigington was on sabbatical at Stanford and during the pandemic, and we were able to get data from University of Michigan in Ann Arbor from the wastewater treatment plant there, as well as influenza incidence rates, because they were doing this study with everyone's COVID samples. They were also testing for influenza, so they had data on influenza occurrence. And then we also were able to obtain some data from Stanford wastewater, and the sports doctor was tracking influenza in the athletes. And so he had some really great data on influenza occurrence. And what we found is that, as you can see, the wastewater concentrations, which varied up to 10 to the 5th copies per gram at Stanford and up to about 10 to the 4th at Michigan, they pretty much matched the incidence rate data. And I'm not showing correlation coefficients, but I think you can see that it matches pretty well. And this was back in 2021 and 2022. So with this data in hand, we felt like we had enough evidence to scale the measurements of influenza A across the United States. And so currently we are providing these measurements, and we have up to 54 months of data for some sites. This is daily data for the last 54 months from Palo Alto Wastewater Treatment Plant. So you can see, you know, every winter we have the influenza peak. Sometimes it's bigger. Sometimes we have this weird summer peak, which I'll talk about later. So this is a really exciting time series to work with. And if we compare, this is for the last 2 winters. This is nationally aggregated influenza A concentrations normalized by PMMOV, which is a pepper virus, which is really common in wastewater. This is for the entire country. So this is over 42,000 digital droplet PCR measurements. And each sample was run in 6 replicates. So multiply that by 6. That's over 200,000 PCR wells that this data came from. And up here is the patient positivity rate from Sentinel Laboratories in the United States. And you can see the really good agreement between positivity rates and influenza in wastewater. Even this really weird little blip right here, it matches that little blip. So one of the things that people are interested in with influenza is, when is the onset of influenza season? Because this would be a really good time to try to get everybody vaccinated. Also, it's important to know the peak, because that might inform hospital staffing and clinical staffing. And then the offset, when is influenza over? And then how long is influenza season? So one of my students, Ilana Chan, who was a data scientist, PhD student, took all of the WastewaterSCAN data up to the point where she was in her PhD to see if we could figure out this information from wastewater alone. So this is actual wastewater data from the previous plot I had shown you, and it shows an example of where we might designate onset or offset, duration, and peak of an influenza season. And the way that we identified onset was when the concentration reached twice our detection limit consecutively for 5 days. And so she looked over influenza season from 2023 to 2024 to see, when did influenza season onset in the different states involved in WastewaterSCAN. And it's interesting because the earlier dates are the darker ones. So influenza season in the US really started in Nevada and Utah, and then it sort of spread across the entire country at the same time. And then the last state up here, I think this is Wyoming. I might be wrong. I should know, right? But that was the last state to onset. So there's this spatial variability in onset of influenza, but at the same time, there's a lot of states that just kind of onset at the same time. And what this plot shows is the cumulative fraction of states in the US that are in onset for influenza as a function of time. And it shows the data for influenza A, which is in pink, influenza B, which is the light green, RSV, which is purple, and human metapneumovirus, which is another respiratory virus. And what's interesting is that you can see that for influenza and RSV, that the cumulative fraction of states in onset went from 0 to 1 over a very short time period. And it sort of shows in another way what I just said, that we had a couple states onset first, and then everything just went up at the same time. And we see that with RSV as well. But what's interesting is that these other viruses have a more shallow slope, meaning the spread is not as instantaneous as with the flu. So that was all interesting to me because we derived all of this from wastewater, but it also matches the clinical data as well. So we showed through this work that influenza A RNA measurements in wastewater are useful for inferring information about disease occurrence, but we also wanted to see if we could infer information about influenza subtypes that were circulating. So could we figure out from wastewater whether H3N2, H1N1, et cetera, were circulating? So in humans right now, H3N2 and H1N1 are the most common subtypes that affect humans. So for this study, we designed and validated digital PCR assays for H1, H3, N1, N2 genes on the influenza genome. And then we retrospectively tested wastewater samples. So this is from San Jose wastewater treatment plant in Santa Clara County in California. And during this particular influenza season, we didn't detect any N1. We detected some N2, and we detected some H3 and very little H1. And this pretty much matched what we knew was circulating in humans from the state of California's sequencing that they were doing, that H3N2, this influenza season, was much more common than H1N1. So this showed us that we could actually infer information about the subtypes circulating from the wastewater. Now, at this time, we asked our our public health partners, would you guys like us to develop an assay for H5, which is avian influenza? And they said, please don't do that. And we said, okay, we won't. But then, of course, we did. So what happened is in spring 2024, which is after influenza season, we actually saw something very strange was happening with the influenza A RNA levels in some wastewater samples. So this is showing the concentration of influenza M gene in wastewater. This is in the 2023-2024 season. And this black line is the national average, aggregated average. And then we saw these really weird things happening at these 3 plants in Texas, where we were seeing these super high concentrations of influenza A in the wastewater after influenza season. So influenza season is over here, and also not similar to the rest of the country. And what was actually happening is that there were a bunch of cows in this area that had avian influenza, but we didn't know it, and nobody else knew it either during this time. It wasn't until March 28th that it was March 25th, 3 days earlier, that they actually confirmed that there was avian influenza infections in dairy cattle. So that was the very first time that they knew that. Yet we were seeing, and it was in this area in Texas, yet we were seeing this in wastewater much, much earlier. So that was pretty curious. So we designed that assay for H5. We used data that had been deposited in public databases, and we developed an assay that was highly specific to the circulating avian influenza clade at the time. And we tested the assay for its specificity and sensitivity in silico and in vitro, hoping that if we detected it, that people would believe that we did. And so what we did is we went to our freezer of biobank samples, and we pulled out those high concentration samples from Texas and also North Carolina, which also in that time had developed some infected herds. And we tested them again for the influenza gene. And we also tested them for H5. And so this on the x-axis is date, and on the y-axis is concentration in gene copies per gram. And you can see at at this Amarillo plant that there was non-detect if you have an open circle here. And then you can see as soon as the influenza started to increase, so did the H5 marker. Same within North Carolina, as well as Dallas, and partially in this other Amarillo plant. These are all in Texas, except for the North Carolina site. So this suggested to us that the influenza we were seeing that spring was from avian influenza in the wastewater. And then we also pulled out some samples from Hawaii and from Gilroy in California, where we knew there was no avian influenza in the cattle and that we did not detect any H5 marker at that time. So this helped us. It was like a counterfactual, another negative control to prove to CDC and others that we were actually detecting avian influenza in wastewater. And looking back at these dates, which were on that previous plot, here is the 4 plants where there were infected herds. And this was the date that they found the infection in cattle. We were seeing H5 in the wastewater before that even occurred. So we scaled that H5 assay in May of 2024 across the United States, despite some people, including the CDC, asking us not to. And since that time, we have detected H5 in only 2.2% of the samples. We have not detected it at all the locations, but in most places where we've detected it, they have identified infected herds of dairy. So the question is, where is it coming from in wastewater? And I have an answer, I think. But first, I wanted to show you a comparison of the H5 data in wastewater and different trends on avian influenza infections. So this is the number of positive samples across WastewaterSCAN over time from 2024 to mid-2025. These are the number of infected herds of cattle, the number of infected poultry flocks, the number of infected wild birds that have been counted, and the number of infected humans. And what we think is happening is that the dairy products that people are consuming in their home that have avian influenza in them, it's just the nucleic acid. It's not actually an infectious virus that is going down the drain and ending up in the wastewater. So it's the infected herds and their dairy product that's causing the wastewater concentrations to be elevated. This is all unpublished data. Hopefully we'll get this paper published soon. So I wanted to talk to you about metagenomic informed influenza monitoring now. So I mentioned this earlier in the plot of the time series from Palo Alto, but there's this really weird situation where this Palo Alto plant had this really high peak of influenza in the summer. So this is the winter peak, and this is the last winter peak, and then there's this summer peak. And we didn't see that in any other location in the whole country. And the levels are quite high. And so the first thing we did is we looked at our measurements of H1, H3, and H5 in the wastewater to see if it was avian influenza. That would be a natural, explanation, because there were not human cases. It wasn't like there were a bunch of people going to the clinics and people had the flu. And sure enough, this summer peak, we didn't detect any of these markers, not H1, not H3 or H5, but still high levels of influenza. Whereas, in the winter peak, we saw a lot of H1 because most of the virus circulating was H1N1 that winter. And so The CDC was contacting us and the local public health departments were contacting us, and we didn't really have an answer. And they sent this email out to some of their partners. We wanted to inform you of a recent spike of influenza A in wastewater at the Palo Alto site. Please see the attached figure. The surge began around July 7th and is really high. Our team has consulted with WastewaterSCAN partners who have no explanation for this. And so we were like, well, that's not okay. We need to help them understand what's going on here. So I mentioned earlier that influenza has a really broad host tropism. It can infect a lot of different animals, but sometimes certain subtypes tend to circulate in certain animals. So we thought if we could figure out which subtype was circulating, we might be able to figure out the source of this influenza peak. So we turn to viral sequencing. So if you want to sequence viruses, there are basically 3 different ways you can do it broadly. One is tiled amplicon sequencing, where you know the sequence of the genome and you amplify all different segments of it and you sequence those. At the other end of the spectrum is shotgun sequencing, where you just sequence everything in the sample and then you look for the viruses. The problem with this is that wastewater has so much stuff in it that if you sequence everything, you get very few viral reads. So we went with this hybrid capture sequencing approach, where there are probes for different viruses that are then help pull the viral nucleic acids out of the wastewater, which you can then sequence. And so we use the TWIST Comprehensive Research Viral Panel that has probes for over 3,000 viruses, and the probes cover almost 100% of the genome of all those viruses. So we went to our samples that were in the freezer and pulled out samples from this peak period here to see what we could find. These are our steps. There's a lot of them, and I won't go through all of them, except to say some highlights here, which is that we did use an inhibitor removal step. We had to use rRNA depletion steps, and we used that TWIST panel I mentioned in Illumina NovaSeq. And then for the bioinformatics, again, I'm not going to go through too much detail, but after filtering and removing human reads and trimming, we did 2 different things. We did read classification with Asvera2 and also some de novo assembly of influenza A genomes. And so this is sort of showing those 2 bioinformatic approaches in one. You get all of your sequences and you look at pre-existing references and you align your reads to help you figure out what you have. And then for the de novo assembly, you take all your reads and you assemble them into contigs, looking for potentially full or partial influenza sequences. And so our read alignment results gave us back that for most of our reads, that they were H13N6, I can't even read that because my vision's so bad, H4, H7, H6, so a bunch of like animal influenzas. But we weren't sure we could trust those because influenza is a segmented virus. And so read classification may not work great. We also wanted to try de novo assembly. And we focused on the HA and the NA genes, which classify the subtypes. And this is an example protein tree of the sequences that we found. So HA are these proteins that stick out of influenza and help it infect the host cell. And this is an example of the HA tree. And we found that most of our sequences belong to H16 in these wastewater samples. And I also should mention, that when we did the same thing with the NA gene, almost all the sequences were N3s. So we're pretty sure we had an H16N3 influenza subtype in the wastewater. And so if you look in the literature, we looked in the literature and found that there is data on this H16N3 virus in migratory gulls in China. And actually, there's a known migration pattern of these gulls that goes all the way down to California, including Palo Alto. So it's not unreasonable to think that these gulls could have been contributing this avian influenza to wastewater. I think the question is, how did it get in the wastewater? Because we have a closed sewage network. So regardless, though, why is this important? I mean, it's kind of cool that we can see this in wastewater and figure out which influenza virus it is. Well, actually, as I mentioned in the beginning of the presentation, influenza has a large pandemic potential and it can reassort. So knowing which subtypes are circulating in animals is important because those subtypes can be contributing nucleic acids that can be incorporated into a human infective influenza and cause more severe illness potentially in people. So having wastewater to help us understand which influenza are circulating both in people and animals is really important for understanding influenza epidemiology and its potential to cause a pandemic. So 5 things to remember from this talk are listed here. So first, influenza A RNA is in wastewater. And patterns of occurrence there can be used to identify influenza season and inform clinical decision-making and public health messaging. Influenza sequences in wastewater can provide information on subtypes circulating in humans, as well as subtypes circulating in the environment. And understanding the circulation of influenza subtypes can allow development of effective vaccines and anticipate recombination and reassortment events, which may change the severity of influenza illnesses. I just have a couple more slides. I want to tell you about some new stuff happening with WastewaterSCAN. So just 2 weeks ago, we scaled the TB assay for Mycobacterium tuberculosis across the United States based on some preliminary data we had that we could detect TB during active TB outbreaks. We have had no detects yet of that, but we're waiting to see what happens. And we also scaled an antibiotic resistance gene, the NDM1, 2 weeks ago. And some example results are below. These are from 3 different wastewater treatment plants, I guess 4 in California. It's kind of interesting that this one has much higher concentrations, but we'll see what we get as we continue to collect the data prospectively. And then perhaps the more interesting thing that we've done is we're scaling the metagenomic work across the country. So we're currently in phase B of our scaling, which means we have 30 sites shown in purple here that we sequence 3 times per week across 21 states. And so these data are collected prospectively and delivered every week. And we put them on a dashboard, which is not public yet, but will be. This is an example of our dashboard. So it shows, this is for Anchorage, Alaska. Sorry, Anchorage, for showing your results publicly. But these are some of the data that we test for. So these are all the different adenoviruses, astrovirus, norovirus, and then chickenpox I put on here for fun. I mean, there are 3,000 viruses, so I can't show them all here. And this shows the reads per million. So a darker purple is a very high level and a yellow is a very low level, and then gray means not detected. And then this is the date here. So there's a scaling bar here to allow you to look at different dates. And there was a pause here between a phase A and phase B. But this gets updated every week. So this had data through mid-June. And so I am scared and also looking forward to trying to interpret all this data, but I will say that we have engaged all of our public health partners who are helping us. In fact, they are helping us design this website, picking color schemes, telling us how they want to see the data so they can best interpret it. So while we're doing the science, we're also interacting with our public health partners to make sure that these data will be useful to them. So these data will eventually be public, but they're not public yet. Okay, so with that, I just want to thank all the people that help with this project. There's a huge team. We work with a biotech lab called Verily to help process the samples. These are some of those people who are so important to this project. The Stanford group, Marlene Wolf at Emory is a co-PI on this project. And then the Sergey Brin Family Foundation supports all this work. So with that, I'm happy to take any questions.
Yeah, I'm thinking that's a really comprehensive and inspiring talk. So I think we, anyway, Professor Boehm and save us even a couple minutes. So we can have around about 30 minutes and for question and answer. So maybe I starting from from the floor, and we also have an audience online. So I would also consider for other like online audience. Maybe I starting a couple of questions from our floor first. Yeah. Okay.
My question is, where did you get the funding, right? This is a new project. So how did you write proposal to any funding agents for your fund?
No, I did not. The project is funded by philanthropic donor Sergey Brin Family Foundation. So they have supported this work with WastewaterSCAN.
Yes, that's always a very good question, and a wishing first.
Hi, Professor Wong. So you did a lot of work, nice work on the viral surveillance in wastewater. So my question is, can we use this data to predict the outbreak from the wastewater? Yeah.
So predict from the wastewater the outbreak? Or can you say it in there?
So for the IVA outbreak, next break from the wastewater.
Yeah. So I think you're talking about a nowcast. So like getting the wastewater data and saying there's going to be an influenza outbreak and it's going to start, like it's starting now. Yes, we can. I think what we don't know is if I can use this data or we can use the data from last season and the season before and the season before to predict when we'll have influenza next winter. No one has worked on that yet. I mean, there are probably people working on it, but this is a great data set to try it because we have so much data from across the country that's been continuous and sometimes daily at plants. So somebody should do it. A lot of this data is public. We publish data descriptors, which have all the methods and then the metadata that go with these data. So you're invited to work on that. Thanks. Let me know what you find.
Okay. So I have one question and one maybe comment. So it is like an epidemiologist's view. So first on your unpublished data on H5 and trying to explain where it is coming from. So the question is, have you tried to check these sources, like different wastewater treatment plants, like where they collect the water from, like from the houses or from the streets? Because my hypothesis would be that it can be that streets, drains, they get water from like wild birds and around, but human waste is human waste. So there can be some kind of distinction. So have you tried to use this like extra data on the water source, wastewater source?
Do you want me to answer your question, or you had a comment too? Do you
Comment
wanna-
on tuberculosis, separate.
Okay, so to answer your question, so in the case of the Amarillo study, which where we first showed we could detect H5, in that study, we actually went up the sewer shed with the help of the local health department and wastewater treatment plant. And we also tested some of these dairy processing facilities that put their discharge into the wastewater, and that had extremely high H5. We also detected off-the-shelf milks from the local stores. Those also had really high levels of H5 nucleic acids, including chocolate milk, which if you ever put chocolate milk in a centrifuge, the chocolate goes to the bottom of the pellet. So I think the way to that sort of suggests like, yes, it's the dairy. There's H5 nucleic acids in cheese that we tested. So we know that we know pretty much that the H5 could be coming from the dairy facilities and also their industrial discharge into the sewer network. You're right, though, that these particular sewers in the US are closed, so we should not be getting urban runoff or stormwater into the wastewater, but we can't rule out that there's cross connections and that wild bird feces could be getting into the runoff and then into the wastewater. So we've been, a student, actually a postdoc, did some modeling to try to figure out, if there was a way to do a mass balance and figure out like it would require so much wild bird feces it wasn't possible or that sort of thing. But yeah, we spent a lot of time thinking about this, and the most clear answer to me is the dairy product. So either people throwing out their milk into the drain or actually the nucleic acid passing through our bodies, because the pepper virus also passes through our virus, those nucleic acids, without being destroyed. So it's possible the H5 RNA could do the same.
I see, but there may be temporal dependency, but doesn't matter. So a comment on tuberculosis. My guess that you will never have any significant reads because of just of the biology of tuberculosis, especially in humans, also in cows, that it really doesn't go into waste. It mostly remains like airborne.
What won't we detect in?
Nah,
What did you say?
I say
We couldn't hear you.
Oops, sorry. Yeah. Very direct. I mean, you likely won't ever find significant quantities of Mycobacterium tuberculosis in wastewaters.
Yeah. Okay, now I hear you.
Yeah.
Yeah, I
Because of
you might be
biology.
right. So we did a study which is in revision under second round of review. Remember the Kansas City tuberculosis? You probably don't remember. In Kansas, in the United States, a couple years ago, last year, there was a lot of tuberculosis in Kansas City. So we actually we have their wastewater, tested it. It was positive for TB, low levels, matched the outbreak, but very low levels, but was there and not there in a plant where there was no TB outbreak. But I don't know. We'll see, right? Because we're going to be testing it. We did detect it before, and supposedly TB cases are on the rise in the US. So maybe we won't detect it. I'm curious. We'll see. But yeah, maybe it's a bunch of non-detects again, and I have to write another paper called Non-Detect Everywhere, which I've done, and we'll do again. Negative results are important. Yeah.
Okay, so I just want to quickly remind our for asking question, identify yourself with a name and yeah. So Gilman.
Hello, Professor Wong. So this is Goldman from Department of Health Technology and Informatics. So a very excellent talk. We actually also working with the public health department in Hong Kong when doing the wastewater surveillance. And we first of all start with the COVID. And then we influenza, and now we also doing the metagenomics. So when we handling the COVID is a bit easier because the concentrations in the wastewater for the COVID is higher when you compare to the influenza. So this is when we dealing with the influenza, the CT value usually quite high, like the 38, 37 around this. And also actually when we dealing with the COVID, the trend usually they will go up a little bit earlier than the clinical case. Usually we have around 1 week or 2 week, earlier than the case. So this is just a very good the early warning for the clinical setting. But for the influenza, we see that the search is actually in the real time with the clinical setting. So people may concern what is the role of the wastewater surveillance for the influenza? Because when you see in the wastewater going up, actually what happening in the hospital? So can you have any, do you have also similar, this kind of the pattern in US?
Yeah, I mean, I think you're right, but I think the thing that you're not thinking about is that actually the data to backfill the influenza positivity rate is delayed like 3 weeks. So if I go right now and I get the data, it wouldn't have any data like until maybe it would have data to June 7th, because the data from June 7th to now is not available or compiled yet. So in real time, that clinical data isn't available. So that's one thing. The second thing is, people don't get tested for influenza very often. So only people who get tested are severely ill people. And so I think we're still learning about like those subclinical cases of influenza and when do they start showing up, and we're still learning about shedding. So I guess I have like a mixed reaction to feeling like the data has to be like useful right away. In no other area of science are we asked that the data we're collecting has like an actual benefit to society like immediately. But so maybe we need more time. But I also think the fact that these clinical data just don't come in, even though you can look at it retrospectively and say, oh, it goes up when the clinical data goes up, that data doesn't fill in for weeks. So
Okay, thank you. Can I have
your question?
the second questions
Yeah,
for that?
please.
So we do have another hurdle about the sequencing part. So actually the nucleic acid in the wastewater is highly fragmented, truncated. So it is perfect, like in your slide, that you can do the de novo assembly, for the typing. Most of the time, actually, in particular, very low concentrations of the influenza A RNA. So it's highly fragmented. What we can do is actually just mapping with the database. And as long as you mentioned the influenza A is a fermented, the genome is fermented. So we are a little bit hesitate to confirm the subtype of the influenza.
Yeah, I think in the example from Palo Alto, it was like all the sequences. We looked at HA and NA independently because they are on the same segment. And all the HA were like 16 and all the NA were 3. So there was one type that was just there. If we had had multiple types, we wouldn't have been able to do that. So I think there has to be a lot more work to do the influenza sequencing in wastewater. And maybe in the end, we'll decide it's not going to be helpful. But actually, even in humans, usually each season, there is a dominant circulating subtype. And our vaccines are made, as you probably know, like a year in advance by a council of experts who are making predictions about next season's influenza. Perhaps the wastewater will tell us were they right or were they not right, and let us know in advance if the vaccine is going to be effective or not. Because we always hear like, oh, this year the vaccine is not effective. Like everyone got it and everyone's getting the flu and really sick. And we might be able to know from wastewater about that. And also, what if we can use the information from wastewater to make mRNA vaccines for influenza, and we can have a really good vaccine that's going to work that season and it just wipes out influenza? I don't know. Maybe that could happen.
Okay, thank you.
Yeah, thank you. So any further questions? Please.
Thank you. That's really brilliant talk. I'm from the University of Hong Kong. I'm also focusing on the influenza and SARS-CoV-2 transmission, but I'm not doing experiments or like wastewater PCR tests. So for the epidemiology field, because I'm not doing those trials and experiments and testing, but I saw that one of your graph is like the positive rate, clinical positive rate is perfectly matched the influenza A concentrations of the wastewater
Yes.
according to your result. But I think that the RNA testing is also contain the both for virus, alive virus and dead virus, right? So I also see that you want to use that data to signal to the time of pandemic's onset, peak period, and offset. I was wondering that if this testing will affect or any delay to your judgment or your result of what the pandemics to the realistic data, like the
Yeah,
public
so I think your question, that's a great question. You're asking about detecting like infectious virus versus just nucleic acids and how that might interfere with the interpretation of the data. Yeah, so I think if you're studying environmental contamination or air, for example, and you're looking for influenza, it's really a big deal to distinguish between those two, because if you want to say, it's really important to know what is the concentration of influenza A infectious virus in this room, and can that cause illness, and lots of interesting modeling to do and stuff. But if it's just RNA, it doesn't matter because it's not going to make you sick inhaling the RNA. In wastewater, it's a different story for this particular application, where what's being excreted by humans is, and once it enters the wastewater, is likely not infectious. It's dead virus. It's just nucleic acids. And so it's just like a biomarker of people who've excreted the virus, or animals, I guess, into the wastewater. So I don't think it matters for this application, but your question is really great because it does matter for other applications of influenza A detection in the environment, like on fomites or surfaces. Yeah.
So you mean it's already like exhalations or output of some infectious individuals or animals?
Yes. So in one of the very first slides I showed was some systematic review data from the literature of people who actually measured concentrations of influenza RNA in excretions in sputum and feces. And when people do have infections, they do excrete it. It wasn't a test of infectious virus. It was just nucleic acid. So if you have the flu, you brush your teeth, you spit into the sink, that sputum is going to have influenza RNA in it. That's going into the wastewater. It's not a indication of like what's airborne or in the air and droplets, for example.
I see. Thank you so much.
Mm-hmm.
Okay. Yep.
Thank you. Yes, I'm Rafael. I'm from the Department of Civil and Environmental Engineering. So, thank you for thank you for sharing your work. It was a very meaningful contribution to the scientific field. But I would like to ask one question about your yes. So, knowing that the animals' wastewater may affect your trend of the data quite significantly based on the summer infectious rate on Palo Alto.
Yeah.
So I would like to ask, even in winter, knowing even though it's very consistent throughout the year, how do you know whether it's from the animals or from the human? How many fractions was it from the animals infection?
I mean, you're asking a really great question. I don't have an answer for you, unfortunately. And I mean, I guess on the one hand, I've been trying to think about like, what is the bright side of animals contributing to wastewater as well? And it's sort of the One Health thing, right? Like if this is a composite sample of humans and also the animals that we live in close proximity to, then it doesn't necessarily matter. It's like the entire animal community's health. But maybe the influenza sequencing will help if we see a lot of animal associated subtypes in the winter. That would be super interesting, along with the normally human subtypes. We'll have that data, I hope, next winter if we continue the metagenomic work through next influenza season. I'm also thinking about like what other zoonotic pathogens can we detect in wastewater. So far, avian influenza is the one that I know about, but I'm trying to think of other zoonotic viruses, or, I mean, we haven't really worked that much with bacteria yet. So that's something that I'm thinking about that I don't know. Do you have any ideas for other animal pathogens that we should look for? I mean, the metagenomic sequencing provides like animal viruses, like, so maybe that will be interesting to look at different animal viruses.
Okay. As we have online audience as well, I just want to entertaining as we receive this kind of questions from online room. So one question asked is how do you accounting for the dilution factor consider with heavy rain flooding? So that's kind of a consider your concentration and the detection of this influenza A virus, because suddenly the dilution factor come over because you report through about whatever per gram or per milliliter type.
Yeah, that's a great question. So I guess first, almost all of the plants we work with are not combined sewer systems, so they should not allow in stormwater. Second is that when you work with the solid fraction of wastewater, I don't know how much solids comes from stormwater. I mean, I know stormwater is really turbid, and so there's probably a lot of solids that could come from wastewater. Third answer is that we do normalize by PMMOV, the pepper mild motile virus, which is like a pepper virus. And so that should correct for the fecal contribution. But the truth is, I think more work needs to be done on this, and it would be good to look at it more closely. Yeah.
Yeah, as we discussed, and my next question is, have you also aware our like a COVID-19 type of wastewater treatment plant? Professor Tong Zhang and came in from Hong Kong U, and also Goldman Shu from PolyU have is running full front. Even Professor Wu and Professor Ben Colling from Hong Kong U Public Health School. So this method can be whatever refer when you design this kind of a wastewater scan program. What can we actually generate more general, like critical or procedures for use worldwide in other regions as well?
Yeah, I mean, that's tricky. I think something interesting I learned during the pandemic, which you all probably learned too, is through this experience, is that everybody has their own way of doing things and doesn't really want to change to someone else's way. And that's okay. But I think wastewater is such a complex matrix, and it's different everywhere and even every time. In fact, I could go flush something down the toilet in my hotel room and completely disrupt a wastewater treatment plant and mess up our COVID testing. So I think I guess what I'm trying to say is I don't think there's a one size fit all approach for this.
No.
But there are some general things that apply across the world, and we all know what those are, and we've talked about them in different, coordination groups and that sort of thing. And I'm working with some colleagues now from AAVOG in Switzerland to write like a primer for Nature. They have a primer about this, and we're going to try to write something general, but it's not anything that all of us doing this know what we're doing and don't want to change. So, yeah.
Yeah, maybe last question from our online audience is regarding the relationship between the rate for viral reproduction and also the spread of the sewage and consider with even environment temperature between your contrasting summer and winter one.
Yeah.
Yeah.
So what's the role of temperature?
The role of the season, compare about the rate for virus reproduction itself, compare about the spread of the sewage, kind of like whatever dilution factor or other things.
Yeah.
Yeah.
Well, that's a hard question. It's multidimensional. I think I'm hoping that the data that we collected can be used by people to explore those kinds of questions. I and my research group can't do everything, which is why we make the data public. So I think it would be great if someone could explore more about the role of like climate and rainfall in the spatial and temporal changes in influenza. We did do something like that for enterovirus D68 and found some really interesting patterns related to the timing of the EVD68 outbreak was related to temperature and latitude, but the duration of the EVD68 season was more impacted by socioeconomic factors like the availability of healthcare. And that kind of makes sense. Like the initial infections in the community were triggered by the environment, but then the like socioeconomic capabilities and resources in the community controlled whether it spread or not. And so that was a real pretty interesting story with EVD68, but we haven't done that with the other viruses. Yeah. I mean, there's so much data. It's really hard to keep up with all this, but hopefully people will be inspired to use the data to look at some of these questions. Okay. Yeah.
Okay. Do we have any question from our floor here? Yes, please. I just want to keep a couple minutes.
Thank you.
Yeah.
My player. The relationship between viral replication rate and temperature in wastewater.
The relationship between the detection rate and temperature? Is that your question in wastewater?
Yeah, I mean it's the relationship between virus
reproduction?
yeah.
Okay. And the temperature.
Yeah.
Yeah.
Replication rate and the temperature in wastewater.
Well, I think there's lots of ways to interpret your question, but the virus should not replicate in wastewater. It should, but I mean, we think that the hotter the wastewater, the more likely the RNA will decay more quickly. Is this your question about decay of the nucleic acids? No.
No.
Or are you talking about R naught, like the reproductive number for influenza? No.
Maybe
temperature is important in the wastewater, and we don't measure temperature. So, but I all these plants, like if I emailed them, they could send me the temperature data, but we don't measure it when we sample. Well, we don't ask them to report it when they actually sample. We don't sample. So
okay,
yeah.
I understand. Thank you.
Mm-hmm.
Yeah. I thinking Patrick first, and then we have Kemi.
Ali, great talk. Patrick Lee from the City University of Hong Kong. Regarding the later part of your data showing the metagenomic data, do you have any evidence of mutation and possible emerging of new subtypes?
Of influenza, no. Well, I mean, we just started acquiring this data very recently, and there's so much to do with it. We have, so I guess my short answer is no, but that doesn't mean that we've looked for it. And we have seen like really interesting results where we have like 100% coverage of like a measles genome, for example. So we are getting like a lot of coverage for different viral genomes. It's just there's 3,000 viruses, and I don't know, like like I said, there's so much to do with this data, but that is one thing that I think would be really important to show, is that you could see the emergence of like a mutation in a virus in wastewater and then see it eventually propagate in the population. I mean, we don't want to see that because that's probably not good, but if scientifically it would be super interesting. And so I think that's an important use for metagenomic data from wastewater.
Right. Because of your longitudinal data set, I feel like mutation is bound to occur. It's almost impossible not to see
Yeah.
mutation over time.
It's going to be a pretty amazing data set when it's complete. Yeah.
Come in.
Thank you very much, Ellie. Really nice to have you right here. Coming Shi from the University of Hong Kong. Let me pretend 25 years ago to be a student asking my professor questions. So it's a layman questions. So you had a sampling in the waste treatment plant, and we understand there are different stage, different phases in the wastewater treatment plants. Can you share some tips about the sampling method? You know, where to sample them more effectively to the data that you think is more representative and useful for your comparison?
Yeah, I think in general, people find that composite samples at the influent of the wastewater treatment plant are the most representative, and that grab samples tend to be noisier. So using like a composite sampler, like an ISCO, to sample flow weighted aliquots, similar to what the regulatory agency might want to test if what the plant's receiving, what the parameters are. Actually, with the solids, so some of our plant, this is in the details, but some of our plants send us composite influent, and some of the plants send us solids that they've collected from the primary clarifier. And those data just get all mixed together. And so those are grab samples from the primary clarifier. And we found that those samples actually are kind of like mock composite samples because of the residence time of the material in the primary clarifier. So, yes, I would say a composite influence sample is probably the best for a wastewater treatment plant. Yeah.
Thank you very much. Thank you.
Okay, so we may have just one or two last question here in the middle. Okay.
Well, thank you for your great talk. I have a question to ask. Because you have two tools, one is the digital droplets PCR, and another one is the metagenomics for surveillance of influenza. I would like to ask, because for metagenomics, it costs a lot of money, and for digital droplets PCR, maybe it can cover most of the surveillance purpose of the surveillance program. Will you have some suggestions? When should we perform the metagenomics surveillance instead of just simply using the digital droplets PCR methods?
Yeah, that's a great question, because the cost actually of you saw all those targets in the droplet digital PCR, and we're doing 6 replicates, is really high, and it's almost as expensive as the metagenomic sequencing. But I think we have been comparing the results from metagenomic sequencing to the digital droplet PCR in the same samples to see, do they agree? Does the relative, like the reads per normalized per the total number of reads for a specific virus, correlate to the digital PCR? Like, could it replace that? And we find generally that it does. For the subset of targets we have data for, I guess the one issue that is the biggest one right now is the turnaround time. For digital droplet PCR, you could get results the next day or even sooner if you have staff working 24 hours. But for the metagenomics, there's so many steps, and the library prep and all that, it just takes time. So at most, maybe 5 days. So we would need to get that shorter. But I think the future is the sequencing. It's just a matter of like if we're ready or not now.
Okay.
Thank you for the insightful sharing. So, my question is on the methodology of wastewater surveillance. So, you have just shared that because virus, be it in the solid phase or the liquid phase, they can't really replicate in the wastewater because it doesn't have a host anymore for replication. My question is, so we know that, surfactants or disinfectants, they could disintegrate the virus, and the virus can naturally break down maybe in a day or two after leaving the host. So were you able to adjust for this underestimation in the sample? Because be it virus or bacteria, they would just, be underestimated in your quantification analysis. And were you able to adjust for that or any, handling process on that? Thank you.
Yeah, that's a great question. So you're asking about the persistence of the biomarkers in wastewater and potentially correcting for differential decay among the samples. And actually, we've done a bunch of work on the persistence of these biomarkers. And to my surprise and shock, they seem super persistent, even because the short fragments of RNA are just not readily degraded. That we target. So that doesn't mean that the influenza A is intact, for example. It doesn't even mean that the entire genome's intact. It just means that short, like 200, 100 base pair target of the digital droplet PCR does not get severed. So it's weird because I always learned RNA is very labile. There's RNases everywhere. Like you have to be super careful. If we throw these viruses into wastewater, just look at natural wastewater with high concentrations of influenza A, it just stays constant at like ambient temperature in the solids for days and days. So we don't correct for that. And I don't have data to support needing to correct for it, at least with the methodologies that we're using, surprisingly. So yeah, thank you for that question.
Okay, so I think in time almost up, and so we just want to thank you, Professor Alexandria and Boehm, to give a really great talk today, and also inspire some of these kind of our students and the staff for future collaboration in this field, and which have a very high potential for public health and also on the environmental surveillance monitoring as well. So we hope can actually building further collaboration with her and with her team. And so please also turn out our pair distinguished lecture series and just join us again next time. Thank you very much. Well, please.